Yes, yes. Well, good afternoon to everybody. Well, in principle, somebody else should have welcomed you, but I had to force Andrea and Silvia to give me the stage first, while I have to leave immediately because there is another meeting taking place 200 meters from here. It's a meeting of ICML together with the Union of Chinese Lymphoma Investigators, which is very important for us. We have a big Chinese delegation here at the meeting. China, with a bit less than 15% of the abstracts submitted to the meeting, is second only to the US. About 25% of the abstracts were submitted by the US, which is, I think, unique for a meeting taking place in Europe. So I have to go there, and then at 3:15 I have to introduce another session at the main meeting. So welcome, and mainly I would like to express the gratitude of ICML to IBSA Foundation, and mainly to Silvia. This is the third time that we have this symposium during our meeting. And this is important and refreshing also in the sense that, well, it is not it is the only session which is not completely devoted only to lymphomaniacs. And I know that some of them are even eager to be here to be listening to something else. So this has been, well, we have about 4,000 people at the meeting and 2,000 more just online, so it has been, it is a good success. Well, let's wait till tomorrow at noon to say the last word, but I think up to now we have been very glad. So I wish you a very interesting session. I don't have doubt that it will be very interesting. I would have loved to stay with you to learn also something more about this topic, but alas, I have to leave. Again, thank you very much, Silvia. Thank you very much, Andrea, as the one who has had first the idea to have during ICML this meeting, and have a nice afternoon. Perhaps see you later at the apéro at 5:30 or something like that in the big tent. Okay, thank you.
Welcome everybody, and thanks for coming. It's an honor for me to have so many people here, very interested, and our special guests and speakers of today. So thanks to Professor Cavalli also, if he's just left. We'll meet him again later. So the welcome through me is from IBSA Foundation for Scientific Research. This is a foundation that was born from the pharmaceutical company IBSA, so that is Institute for Biochimique, based here in Lugano. And almost 10 years ago, the president of the company decided to have a foundation like this to support scientific research. And so we are dedicated to support through events like today, through our activities of fellowships, scholarships, and also a lot of projects and events more dedicated to outreach, so to dissemination of scientific culture. So my role today is just to thank the people that work with me. The collaboration with Professor Cavalli, with this meeting, as he said, is started many years ago. So each time, every not year, 1 year, every 2 years, when there is this meeting so important for Lugano, we have always a special session together, like today. And so this is, in this way, we have the possibility to invite very special top scientists as what we have, the people that we have today. So at this time, I would like to introduce Professor Andrea Limonti. Andrea is a famous scientist, professor almost everywhere. But the most important thing is that he's a member of our scientific board. So I leave the stage to him, that he'll introduce the topic and the speakers. Andrea.
So thank you. Thank you, Silvia. So it took me more than 5 years to convince Silvia that we should spend the money to invite eminent scientists and to have such a beautiful event, right? So, distinguished guests, ladies and gentlemen, Margaret Foti, President, CEO of the American Association of Cancer Research, good afternoon and welcome. So, it's a pleasure for me to introduce this IBSA Special Symposium that takes place normally every 2 years during the ICML Lugano meeting, which is the largest lymphoma meeting in the world. So the meeting is organized by IBSA, but also by the Institute of Oncology Research, that is a translational research institute here in Switzerland that is devoted to the identification of novel therapy. So today, I have the pleasure to introduce 3 eminent speakers. So the first 2 speakers, Hans Clever and René Bernard, are worldwide recognized as leaders in the field of cancer biology, innovative therapy, whereas the last speaker is a very young talent investigator, Arianna Baggiolini, that will deliver a short presentation but extremely interesting. So the forum will focus on personalized medicine oncology, this is the title, and in particular on novel modality to study cancer biology, identifying combination therapy for the benefit of cancer patients. So by saying that, it's my pleasure to introduce the first speakers of today's section, which is Professor Hans Clever. And I have to do a small summary of his amazing CV, so it will take 30 minutes to do it, but I will try to be very short. So Professor Clever is an MD PhD and is currently the head of pharma research, PRED of Roche. He's also a member of the corporate executive committee of Roche. Professor Clever has led for almost 20 years a very big research group of more than 30 scientists at the Hubrecht Institute of Developmental Biology, and is also still a full professor in molecular genetics at the University of Utrecht. So Clever is recognized for his contribution to the field of cancer biology, many different discovery, Wnt signaling in colon cancer, beta-catenin, and so on. And recently, it's very well known, particularly for having pioneered the development, the use of organoids to model the biology of different cancer. He has published more than 700 papers, and his contribution has been recognized with several prestigious research awards. Outside academia, he's also created different startup companies which are very successful. So the title of his talk is Organoids Model for Human Disease. Professor Clever, this floor is yours. Thank you.
Sorry, go back. Yeah, now it works. Yeah, well, first of all, thanks very much for the invitation to speak for the IBSA Foundation. Also, thanks to Andrea for a kind introduction. So what I'll try to do in the next 30 or 40 minutes is give a very general background on how my lab, about 15 years ago, totally by chance, by serendipity, developed a version of organoid technology, and then I'll give you a number of examples as to how we have been using organoids, focusing mostly on cancer and personalized approaches in cancer. So what you see here is the intestine of the small intestine of a mouse or of a human. The inner lining of the small intestine, we read about this 30 years ago, is probably the most rapidly self-renewing tissue of a mammalian body. So in a mouse, every 5 days, the entire inner lining is replaced by the action of stem cells that were then unknown. And the structures that you see here are called the crypts of Lieberkühn. Those of you who've studied histology probably know that term. The protrusions that stick out into the lumen of the guts are called villi. So villus and villi, I'll use those terms. And then the crypts are the ones that stick out in the walls of the gut. And these multicolored cells here, I'll try to convince you in the beginning of my talk that they are actually the cells that drive this incredibly active self-renewing, physiological self-renewing process. So here you see one villus, a few more villi in the background. So that's the inside of the small intestine. You see two crypts. There are probably about 8 or so that surround the circumference of a villus. Stem cells somewhere near the base of the crypt. They were known for a long time. Produce daughter cells, and these daughter cells were shown in the 50s already by Leblanc, so almost a century ago, in Canada, to produce daughter cells, and these daughter cells divide for 2 days while they move up. You see them moving up in the crypts. After these 2 days, where they cycle every 12 hours or so, they will then pick up one of about 12 different cell types, and I think I have an image here. So people have thought of this very much as the way people think about stem cells in the hematopoietic system. So a single stem cell makes all these different cell types, and as I say, by single cell sequencing, we now know there are probably about 12 or so of these cell types. So you'll see most of them on the flanks of the villi moving up. The pennant cell, the one in yellow here, actually moves down and settles at the base of the crypt. They live slightly longer than all the others, but all other cell types will continue to move up, and by the age of 5 days, they reach the tips of the villi and they die. So every 5 days in a mouse, probably a week or 10 days in the human, this tissue lining is entirely replaced, and in man, you're probably talking about 100 to 200 grams of tissue that are produced every day and that are destroyed maybe a week later. So we already mentioned by Andrea a long time ago found a few components of the Wnt pathway, and when we knocked them out, and Wnt signaling was then entirely in the field, in the realm of developmental biology, so key in thousands of millions of fate decisions in developing embryos, flies, worms, mammals, you name them. But to our surprise, when we knocked out one of these components, TCF4, we saw that the process that I just showed to you totally stopped. So that implied that whatever the stem cells were, they require Wnt, and when you block Wnt genetically in mice, this process stops, and actually 5 days later these mice die because all of their villus is gone, so they can no longer take up nutrients and, more importantly, liquids, and they basically dehydrate and they die. So Wnt was crucial in an adult. We now would probably feel safe to say that almost every adult stem cell type will require Wnt as one of its key drivers of its activity. And then at about the same time, in the collaboration with Bert Vogelstein, who had been working on a gene called APC for a long time, I get back to that a bit later in my talk, we could also show that mutational activation of the same pathway, of the Wnt pathway, leads to colon cancer. It's actually the first mutation that will lead to the formation of a polyp, and then with some sequential mutations, I'll show them, you'll get a full-blown invasive and metastasizing colon cancer. So there were these two things. Wnt is physiologically important to drive the stem cells, and in cancer, it's the same pathway that is mutationally activated. And then when single-cell sequencing wasn't available, of course, but when DNA arraying became available in 2000, invented by Pat Brown, we could then ask, what are the genes that are controlled by the Wnt pathway in cancer and in crypts? And by careful comparison of those lists, actually they were very similar. Nick Barker came up with I hope you can see the slide well, but came up with a marker that essentially had not been studied very much, LGR5, a 7-transmembrane receptor, that when he made a knock-in mouse here with GFP, lit up in green these tiny cells sitting between the much darker cells, the larger cells at the base of the crypt, called the Paneth cells. I briefly mentioned them. And so we thought originally that we identified a novel cell type, but then we realized that they had been seen 50 years earlier by LeBlanc and actually been postulated to be the stem cells of this system, and then sort of forgotten. And so we revived his theories. Now with the technologies that were available, we could actually prove that there are the stem cells. So the mouse that has a GFP in the LGR5 locus, so the stem cells are green, also have a recombinase that we can activate, Cree recombinase, in that same locus. So the green cells we can now activate to become blue, explained in a simple version. And if you do this in an adult mouse and you only hit a few of the stem cells, what you'll see is a ribbon, blue ribbon forming. So within 2 days, the first dividing daughter cells leave the crypt. They form this, you saw the major cell types there, goblet cells, enterocytes. Day 3 and 4 and 5, they continue to move up the flanks of the villi. And when they're about 5 days old in the mouse, so again in the human this is a bit slower, the first blue daughter cells reach the tips of the villi and they undergo apoptosis. Now, if you don't analyze the mice now, but we wait for 2.5 years, we still see these ribbons, implying that that cell that we marked in blue is long-lived. Doesn't live 5 days, but it lives as long as the mouse. And also, if we do the experiment well and we mark only one cell in a crypt, we can then read out what it makes. And actually, all the other 12 cell types will be present in that ribbon. So essentially, the cell is multipotent, long-lived. We proposed it to be the stem cell. There was a lot of resistance originally because they didn't really look like the stem cells from the textbooks. And most importantly, they constantly divide. And actually, being quiescent was being used widely in the stem cell field to identify potential stem cells. You label DNA, rapidly dividing cells diluted out. Any remaining cell that keeps that DNA label would be the stem cell. Now, these cells divide every day, and in a mouse, they never stop. And they go through about 1,000 consecutive cell divisions. A single cell will do that. So there's a different way of doing this experiment using the confetti locus. We obtained a multicolor cassette from Deb Lickman in Boston, who've used this cassette to mark events in the brain. We rebuilt it, made another knockout mouse that we called confetti, and this now allowed us to label multiple stem cells, each in their individual colors, using the same tricks. And actually, when you make it homozygous for the geneticist, you have 10 false colors, so you can essentially look at every individual stem cell. And Jeroen Huiber, who makes these movies, predicted that this would be the outcome if we would use confetti to basically to do a lineage tracing for stem cells. And that turned out to be the case. So here you see a low magnification image with a larger magnification. So eventually, this allowed us to study the kinetics. I won't go into detail of these stem cells, but if you wait long enough, a single stem cell in each crypt will win, and then the entire crypt will be blue or red or yellow or green in the 4-color version. And for the rest of the lifetime of a mouse, you get these parallel bands of colored cells that don't mix very much and run to the tips of the villi. And again, this will last for the lifetime of a mouse. while Nick was doing the experiments, we realized that these events were not only playing out in the gut, but also, for instance, in the stomach and in the skin and in hair follicles. And we started realizing that LGR5 was probably a marker for many more unknown and known stem cells. And I think I would now safely dare to say that LGR5 is very likely a marker of every active epithelial stem cell type. So being it ectodermal, mesodermal, or endodermal. I won't talk about the function, but that was also meanwhile solved simultaneously by a number of labs, including mine. But it turns out that LGR5 allows our spondins to amplify Wnt signals. So it actually plays a role itself in strengthening Wnt signals such that stem cells can divide. Anyway, so we didn't know that our spondin was the ligand of LGR5. You see the terms in the top here in the title. But we knew that our spondin was a potent activator of the Wnt pathway, an amplifier of the Wnt pathway, I should say. So when we realized that these stem cells were not behaving as any other stem cell that was known in terms that it constantly divides, Toshi Sato in the lab and some others sat down and we started thinking about ways of maybe take one stem cell and make many stem cells out of them. And the idea would be we provide the growth factors that you would normally see in a crypt. And we knew quite a bit because we had been for 10 years randomly knocking in and activating all sorts of pathways to learn a little bit more about what a stem cell in the crypt really needs. And that's Wnt, I already told you. Epithelial growth factor was widely known to be a mitogen for these epithelial cells, and we needed to block noggin. We needed to block BMP signals, also known from a transgenic that we sort of randomly made a number of years earlier. And the combination of these three recombinant proteins, so activating, amplifying Wnt through our spondin, which we now know works through LGR5, we didn't know it then, EGF, tyrosine kinase receptor activator, and a BMP inhibitor. That actually is also the cocktail that works for almost all other tissues. You usually have to add more, but these are the three basic components of the cocktail. There's no serum, there's matrigel that is key, so laminin and collagen, we grow them in 3D, so they don't stick to plastic. As soon as they touch the plastic of a plate, they are gone. But we see these strange, or Toshi sees these very strange structures appearing. And when we looked more carefully, they could actually merge, they could fuse. If you put them in collagen gels that have long bundles, and Norman Sachs, who did this in the lab, could create tubes that were like 2 to 3 centimeters long, and it started looking more and more like a real gut. And looking very carefully, the buds that stick out of these organoids look like crypts, so that's where the stem cells are, the pennant cells that I mentioned, the proliferating cells, and the inner lining of these cystic structures essentially have all of the other cell types of the gut. So what the stem cell, with very few cues, only 3 growth factors, nothing else, what it does, it tries to recreate its original tissue. It's really surprising. We're still studying how it does it. We don't really understand it very well. But that, of course, was a big surprise. And more surprises were that actually these never stopped. So we've now grown them, these mouse organoids, for about 12 years. Toshi originally dubbed them mini-guts. I think the term now would be gut organoid, because you can make them now from many other tissues. Telomeres stay long. There's no senescence under these conditions. Particularly Wnt seems to counteract senescence. And we've also importantly never seen by sequencing oncogenic transformation. So one immediate reaction would be, okay, these cells grow for so long, they must somehow be transformed. Actually, this is a more recent version from human small intestine, where you can also see all of the cell types are represented, and they're actually very amenable to CRISPRing. So in this case, Hui Vi He and Lin Lin knocked in an orange marker in defensin A, which is a pennant cell marker, A5, MUC2, a goblet cell marker in green, and chromogranin A, a hormone secreting the enteroendocrine cells of the gut. There's 6 different types, but they're all marked by chromogranin A. And you can see they did 3 knock-ins, sequential knock-ins, and we can now see all these cells in a live organoid happily functioning. And now, for instance, the enteroendocrine cells, but also the M cells and the tough cells and the best cells could never be studied. There are no cell lines, and mice typically don't have these cell lines, or these cell lineage, or they do very different things. But these organoids now allow us to probe biology, human biology, in a way that was previously not really possible. So how normal are they really? This is also an old experiment. Mamoru Watanabe in Tokyo was interested in seeing if they could be transplanted. And then together with with Toshi Sato, who is still in my lab then, who's now a professor at Keio University, Toshi grew up a single mouse stem cell to about 100 million cells, froze them, sent them to Tokyo in the form of these organoids, and he made them from a mouse where every cell is red fluorescent, so it's expressing RFP for the insiders, and that would allow us actually to recognize these cells, because normally, as you know, a mouse is not red fluorescent. In Tokyo, Mamoru's people had treated mice with DSS, which is a chemical in the drinking water, a very popular model for inflammatory bowel disease. These mice developed ulcers in their colon, and they infused our organoids, red fluorescent organoids, into the lumens of these colons. And these organoids will float around. They typically have their integrins on the outside. So when they sense collagens or laminins or other sub-epithelial structures, they'll actually land, and like a living Band-Aid, they seal these lesions. And actually it works very well in a mouse. I think the experiment is here. So on the top left you see a mouse colon. What is black is the recipient Japanese mouse, and what is orange-red is the offspring of this one stem cell. So it basically was one stem cell that was used for several dozen mice. And actually you could have probably transplanted several thousand mice, because there appears to be no limit to the expansion potential of these cells. And then if you want to find them back, you really have to look for the fluorescence, the red fluorescence, because in any other means, these are normal cells that integrate fully normally and seem to have all the normal functions of the adjacent Japanese epithelial cells. One prediction would have been, well, no, they've grown so extensively, overstimulated, they'll now transform somehow, they'll form polyps, something like that. That has never been seen. Also, we never see oncogenic mutations in these cultures. Other prediction maybe from the hematopoietic stem cell field, where if you let hematopoietic stem cells divide too much, they exhaust and then they disappear. But these you can apparently culture forever and they never stop. And that's why we can start from one cell. And while this was all ongoing, Nick was realizing that LGR5 actually marks other stem cells, other tissues, and Toshi started trying to culture those as well. He started with pancreas, but there's now a long list of epithelia. I should say it's really restricted to the epithelial parts of organs. If you think about our internal organs, the epithelial cell types are the ones that mediate the organ-specific function. All else is blood vessels and nerves and immune cells and stroma, important, but not for the dedicated function of that organ. And you can do this essentially with playing around a little bit with conditions, culture conditions, particularly growth factor cocktails, for any tissue that you'd like to try. And this can, of course, be used for any sort of basic scientific experiment, but also you can do this directly from patients or from individuals and address personalized ways of treatment or of disease progression. And we and many other labs now have used these models to study cancer, to generate models from hereditary diseased patients, and it's also very nice for infectious disease. And there's some very good stories that I unfortunately don't have the time to talk from other labs. I'll show you just one. So huge difference with cell lines is, first of all, these cells are normal, but more importantly, all the differentiated cell types that you would see in an organ will ideally also be present in this organoid. And there's a fair number of parasites and viruses and bacteria that cannot be grown in the lab because they don't propagate themselves on cancer cells. Essentially, cell lines are almost always cancer cells. And one good example is norovirus. You probably have all heard of norovirus. You might have gotten it. It's a terrible virus for 1 or 2 days. That could never be grown in the lab. Maria Estes has published that actually you can grow them on normal human small intestinal organoids. You have to add a little bit of bile acid, and then the virus will grow. And it uses the differentiated cells that are present in organoids. Cryptosporidium, that I'm just showing here, does the same thing. It's a protozoan, so a eukaryote closely related to malaria that goes through a complex cycle. Actually, it causes chronic enteritis, particularly in lower income countries, very hard to treat. And again, no lab model that allows one to study this virus. And this is again a movie, so I'll show you the experiment. So what we hope to do, you can actually buy the starting stage called the oocyst. You see it here, produced in cattle in Texas. For some reason, when you buy them, you can infect other animals with it, but we tried to see if you could actually propagate these oocysts inside human organoids. And the idea would be that the oocyst will find the cell type that they can infect, because all of the cell types are there, and that actually this would then allow them to go through this entire asexual and sexual life cycle. And what you see here is the oocyst. So there's 4 sporozoites in a single oocyst. You see the sporozoites coming out here. They're now looking for a cell to infect. It turns out they really love the enterocytes, and this is about 90% of the gut epithelium. These are the cells that take up the nutrients and the liquids. So they infect. Then you can actually recognize the different stages because they all have different numbers of nuclei. So this is now 8, the type 1 merozoite. So it's an amplification step. They go again, look for a host cell. Again, it's an enterocyte, never present in cell lines, but abundantly present in organoids. Make a type 2 meron. So we're now about a day into the life cycle of this thing. Now they enter the sexual stage. So they'll produce a so-called macrogamete, which would be the ovary, and a microgamete. I think the macrogamete is in the back, yeah, and the microgamete is in the front. So the microgamete produces sort of spermatozoa-like little things, many of them. This is the lucky guy that actually managed to fertilize in the microgamete the equivalent of an oocyte. Divides 2 times. Now you have 4 nuclei, and the circle is round. Now it releases itself, and there are 4 sporozoites inside. So that was the hope that we would see this, and indeed that's what we saw by EM. We can essentially see all of these stages of this parasite. And actually, when we take these organoids, we can infect mice when we extract the oocysts produced there, and this essentially shows that we have completed the life cycle. And Matthias Lutolf then in Lausanne has shown actually you can using similar systems create a situation of chronic infection of many months where you constantly every day they produce these oocysts, much like what would happen in an infected animal or an infected human being. So just one example. The rest I think is all cancer. When we realized we could grow essentially all epithelia, the implication would be we can grow carcinomas, because carcinomas by definition arise in epithelial cells. That turns out to be the case, but there are actually some complications that I spend a few words on. But in essence, what we can do from an individual patient, we can get a biopsy from flanking or healthy tissue, and we can get a biopsy from the tumor, and we can grow them. We can sequence them. Everything in the tumor sample will die. Everything that's not a cancer cell will die. So immune cells will disappear, blood vessels, all that. So it's not a tumor, it's a cancer organoid. But the attractive thing here is that you have the live cancer cells. And there are now, so I'll show this a bit later, but there are now many, many studies showing that if you test drugs on these in a personalized fashion on these organoids, that this has a very high predictive value of how a patient will respond to the same drug combination. Challenges are that it's very slow, it's very expensive, you need dedicated personnel to do this, so it clearly needs automation, miniaturization, using microfluidics, probably AI-driven image analysis, etc., but the principle is there. There are now large biobanks. Many labs have done this for many different types of carcinomas. You see them here. Brain tumors, we've started being able to culture those as well. They require very different growth factors. So all epithelia need Wnt, tyrosine kinase receptor activator, BMP TGF-beta inhibitor, some stuff on top, but all others, like the hematopoietic stem cell, people still, I don't think, know how to expand that long-term in culture. This is an example, normal and tumor. And then the first paper came out in 2018, which scooped my lab actually. Beautiful paper in the UK. 5 or so small phase 1-2 trials where in parallel organoids were grown and patients were treated. Organoids were tested with the same drugs that were in the phase 1-2 trial. And actually the outcomes claimed in this paper is indeed the correct predictions in the order of 85 to 90%. So if the organoids tell you the drug will not work, 90% or so of the cases, that prediction is correct, and vice versa, if it does work, you'll also confirm that. Then there's in 2019 a few more papers where we have some involvement, and now there are probably 30 or 40 studies that keep on confirming this. Having said that, immune oncology cannot be tested in this setting. There's no immune component here, and also angiogenesis inhibitors would not score here because these things are not dependent on blood vessels. A complication that's very unexpected is actually that under these conditions, the normal tissue grows faster than the cancer tissue. That's counterintuitive, but if you make movies, this is actually normal from an individual, normal, healthy tissue. And when you make movies, you'll see them here, you'll see cell divisions at a certain rate, and every cell division results in 2 happy daughter cells. And the tissue essentially just expands exponentially. If we now look at a cancer organoid from the same patient, colon cancer, you'll see that, well, first of all, almost always aneuploid, and you'll see that they have a very hard time to basically to divide and then end up with 2 healthy daughter cells. And you see, for instance, here in the lower bottom, you see attempts, probably 100 chromosomes, 2 nuclei in 1 cell, big mass, and on the top, I think these will die. On the bottom left. And if you follow these, so the attempts of trying to go into mitosis occur at the same rate, but they're just not so good in completing mitosis. They run into checkpoints, or the cells die, or one daughter dies. And this actually means that when you have a mixed sample, so we start organoid cultures usually from a small biopsy, when that's a mixed sample of healthy tissue and cancer tissue, the healthy tissue invariably grows faster than the cancer tissue, and after 3 or 4 passages, 3-4 weeks, you're left with healthy cells rather than cancer cells. So very counterintuitive, now seen by many labs. And a complication, for instance, prostate cancer typically is always mixed. Healthy and prostate cancer cells are in the tumor completely mixed, so it's impossible to get a clean biopsy. And I know of no lab yet that actually can grow prostate cancer organoids long term, because they always end up with normal prostate, and you can only do this from metastasis where there are no normal prostate cells and only prostate cancer cells. For other cancers, there are tricks, like colon cancer always needs wind, and I'll show how we use that. And colon organoids always need wind. And when they're colon cancer, they no longer need exogenous wind because they have the mutations, the activational mutations in the pathway. One way we can use it, so the first small biobank that we made, colorectal cancer, about 25 patients or so. The 6 on the left are the MSIs, the hypermutated, and the ones on the right are the MSSs, the non-MSIs. And there was one sample in this, so one patient who in their tumor had no mutations anywhere in the EGF receptor RAS pathway. And that's patient 18, you see a green box here. So we could then create an isogenic pair where we took this particular cancer that essentially represents a single patient, and we added on top of all the mutations a KRAS mutation, and this was done together with Hugo Snippert, former PhD student who has his own lab in Utrecht. And when we now expose them, and I don't know exactly which inhibitors they are, but to a mix of EGF receptor and MEK inhibitors, quite popular, being investigated also for colon cancers. The cell line itself, the organoid line on the left, you can see it fragments its nuclei. It's very effective in killing these organoids. But now we just add a single base change, so we add a single G12D mutation on the right. What actually these cells do, they will, when the compounds are there, they will stop cycling, but they stay alive. And the moment you take the drug away, there is a burst of proliferation. So this is known, this phenomenon, but it's a very nice, I think, demonstration that these organoids can really be used, in a detailed way, the way people have used mice, to actually study the effects of individual oncogenes. Another study of a number of years old, but done independently in my lab by Jarno, now independent in the Maxima in Utrecht, and Toshi Sato, then already a professor in Keio University. We both realized, without having spoken between the labs, that the common mutations in colon cancer, the most common ones, you see them here, APC, KRAS, MET4, and P53, exactly reflect the growth factor dependencies of our organoids. So So empirically, we had found that a normal colon stem cell needs Wnt are spondin, you see it in the top there. It needs EGF, and it needs noggin. Now, what the cancers do, they mutate APC, which is a negative regulator of the Wnt pathway. They mutate KRAS, which takes the dependence on EGF away, theoretically, and they mutate SMAD4, which is a transcription factor in the BMP pathway, TGF-beta pathway. So if they lose SMAD4, they also no longer need an outside inhibition of the BMP pathway. So really rather surprising that what the cancers do is exactly what we had empirically found out to keep the cells growing. We actually keep the cells with exogenous growth factors. We keep them normal, benign. But when you make the mutations, you essentially, that was all hypothetical, you turn them into a cancer. Now, what you see here at the bottom is from a paper from 1990 by Eric Vieren and Bert Vogelstein, who then proposed, without knowing all of the genes involved, that colon cancer would arise by a sort of ordered series of mutations that would take a normal cell on the far left through these early, intermediate, and late adenoma stages to an invasive and metastasizing carcinoma. So what we hope to do by using CRISPR that had become available in organoids, that we could recapitulate, much like you would do by crossing mice, recapitulate the formation of a human cancer. Again, captured in a movie. So what we hope to do is obtain normal tissue. So again, we don't start from a stem cell, we just start from a little bit of tissue, put it in culture. And what we hope to do is recreate this adenoma to carcinoma sequence as proposed by Vieren and Vogelstein. And at the same time show that this idea that the mutations of colon cancer essentially reflect the growth factors that the normal stem cells need, we would also prove. So we start with it targeting APC. Theoretically, now they should be Wnt independent, so we leave Wnt out of the medium. You see the growth factors top right. And indeed, we get single clones growing out, and they have lost both alleles of APC. P53, we can actually take out with CRISPR again, and then we can select by adding a small molecule, actually, I think it's a Roche molecule, Nutlin. So if we kill the wild type P53s, it's like an anti-cancer drug. 2 mutations. KRAS, we knock it in, leave EGF out of the medium. So now only the KRAS mutants will grow, and they will have now 3 mutations, and they only require noggin, the BMP inhibitor. And then finally, we target the 4th gene, SMAD4, transcription factor in the BMP pathway. Cells can no longer transmit BMP signals. Leave noggin out, and now essentially the only thing that can grow is an organoid that has all 4 mutations. And you can actually do this in any order. You can also do it all at the same time. We've now done 6 or 7 at the same time. Select with Nutlin, so every organoid you get is P53 mutant, but then it'll have a mix of the other genes you try to target, so you make a small library of all sorts of combinations of the genes of interest. And then finally, the readout would be just looking at these cells, but you can also transplant, and we do that orthotopically into the colons of immunodeficient mice, and you see the outcome here. Only the ones with the 4 mutations grow in a way that our pathologist calls a human colorectal adenocarcinoma. It invades actually very actively, and it will metastasize almost immediately to the liver, where in normal humans it would also go. And all the other ones, at best, give an adenoma or something that essentially doesn't grow, or something that we couldn't even find back after transplantation. Yeah, the movie doesn't play, but I'll say what you could have seen here. So what you see is in yellow is bad, you can't activate it. Okay. So what you see in yellow here are breast cancer organoids, and this is a study that was recently published just a few months ago from Anne Rielse's lab mostly, together with Jürgen Kubal in Utrecht and his company Gadeta and my lab. And so what this is essentially in blue-white you see CAR T cells, and these CAR T cells have been transduced with an unusual CAR T receptor, namely a gamma delta T cell receptor that's not modified. It's a V gamma delta 9, V delta 2. There's quite a bit of literature on this receptor now, and it somehow sees the difference between a transformed cell and a normal cell. It's in trials for AML and multiple myeloma, but actually we could not believe this, but when you take these cells and you throw them on the healthy colon organoids, they will not kill. When you activate KRAS, they will kill. So they are believed to see this molecule BTN2A1, directly, so there's no MHC presentation for the immunologists, and they'll see a conformational change of this molecule that signals that the cell that is carrying this marker is hyperactivated, likely a cancer cell. Spectacular thing. Now, what we did, and unfortunately you can't really see how this works in action, but you see here breast cancer organoid in yellow. When the breast cancer cells are killed, they turn red. And white blue are these CAR T cells. So they're typically, 4-week cultures transduced with these gamma delta T cell receptors. They are the standard CD4, CD8 cells you'll find in peripheral blood. And so Anne basically takes these movies over 24 hours, and then using machine learning, she classifies the behavior of the T cells, and they behave very differently. So some of them do nothing, others run around like crazy, but they don't kill, others will touch organoids, but nothing else, others will kill. And then so then she classifies them into about 6 or 7 different behavioral classes, and then you can zoom into these files and see what an individual T cell does. And this is actually quite spectacular. And I should say it's not from my lab, it's from Anne's lab. So here's one of these cells. So it engages probably about an organoid of probably 20 of these breast cancer cells. 11 hours in the experiment, you can see it wraps itself around the organoid, and one by one it kills off the cancer cells in a matter of a day. I don't think this has ever been seen before. This cell essentially destroys the entire organoid. So destroys, I'm almost there, destroys all of these cells. Another detail here, so this one from the left sticks out these very long extensions. They call them, Florijn and Anne call them sling and lassos. You can already see it wraps around cells. This one also in a matter of, there's a friend helping, but this one also managed to kill the entire organoid, probably I would estimate 30 or so cancer cells. So a single T cell can do this. But the most spectacular one is this one, is my last slide, I believe. It's called the serial killer. So this is extremely active. We're now 2 hours in the experiment, already killed 10 cells. You see it drills through these organoids, runs away. There's probably chemotaxis involved because it looks like it knows where the other organoids are, kills a few more. We're only 5 hours in the experiment now, runs off to a third organoid, kills more of these cancer cells, and then disappears into the sunset. Now, these are the ones that you really like to have, and when you give them back to a patient, I would assume they're about 2 to 3% of the cells in a typical preparation. And the way to isolate them is to spin the organoids down now, so 2 hours after they engage the organoids, they're inside. You then do a single cell sequence. It's all in this paper that you might want to look up. And so we now have a signature. Anne now has a signature of about 50 genes that are uniquely expressed only in these extremely active CD8 killer cells. Quite a few of them are known markers, but also novel markers there. And with these markers in hand, you can now actually purify these and maybe give back far fewer CAR T cells, but give the right ones, or maybe use them to guide the culturing of these cells and create many more of these types of CAR T cells. And with that, yeah, I think I named everybody along my talk, and I thank you very much for your attention.
Thank you. Thank you. So thank you, Hans. Beautiful talk. I think we have 5 minutes for some question. Would be great if some student ask a question to Hans. Otherwise, I would be forced to. Oh yeah, we have a question there. Not a student.
Paul Meijners from Antwerp. You said that you were a little bit surprised that the normal tissue cells took over the cancer cells. But could it also be that is the normal situation, that actually that the cancer succeeds in getting in is exceptional?
Yeah, I think that, I think there's 2 things. First of all, I think that is true. So it's a way of getting rid of potential cancer cells early on because they are slower dividing. But this would only work in a rapidly dividing tissue, and it's like the gut. Tissues that don't divide, they have all the time in the world to survive this. I think another implication might be that So my lab calls the cancer cells now the zombie cells because they cannot sprint. They sort of just stumble along and pieces fall off, but they eventually will get you. Now, the reason is that it's probably much easier to push them over the edge because they are not so fast growing as normal tissue. So even if you would kill the normal tissue off rapidly with your chemotherapeutics, which is what we see, so almost no cancer drug is specific for cancer cells when you compare cancer to normal, but the normal tissue recovers much more rapidly. So if there's just a few cells left in a crypt, they grow a crypt back in a matter of 3 days, 4 days or so, much faster than a cancer would ever grow. So there might be one of the reasons why classical chemotherapeutics actually do work.
There is another question there, another here.
Hi, Alexander Stamers from the Prins Maxima Lab, Centrum in Utrecht. I got a question about the healthy organoids which you transplanted into the mice, and you saw that they actually stopped growing once you transplanted them. Do you know any idea why they stopped growing?
Yeah, because so in culture, you have to realize the situation in culture is really not a normal situation. It is a situation in which you basically activate maximally all of the pathways that allow a stem cell to grow as fast as it can. So it's like a damage response situation. But they can maintain this for long periods of time. The moment you take them out of this culture medium, or you just reduce the growth factors, they will actually slow down and they fully differentiate. So the moment you transplant, they apparently recreate crypts, and that's where the local signaling is good enough for stem cells. But just a little bit higher up, there's no wind anymore, and so the cells that move up naturally will differentiate out. And so cancers will, at the base of the crypt, they look the same as a stem cell, but while they would move up in a structure like that, they would not switch off those pathways, and that's why they form a polyp. But it's just, we can mimic this quite easily in vitro. So most tissues under high growth factor conditions will rapidly grow and be rather undifferentiated. And if we really want to have all of the cell types there, we have to take the growth factors out. And then you see, and we can steer a little bit by changing growth factors which direction they go. Like Notch will do that. But yeah, it's the normal behavior of all of these organoids.
Thank you.
Another question here.
It's futuristic, it's incredible. It's meant to treat the patient one by one based on the model of the disease at that point of the day. And that's great. My question is, would it be possible, for example, to have a patient that has a complete response to the treatment and to have also to model him on the animal in that case, and then to foresee what are the prognostic of relapse? That will be very nice to know more about that.
Well, there's a number of studies that address this, so maybe what I can say is that if you have enough cells, so if you would have 10, 20, 30 million organoid cells, and you start treating what looks like a complete response, so everything dies, a week later clones come up that are resistant. And actually, it turns out they're already present in the original biopsy. And so you would probably in the patient call a complete response and then a recurrence later. But in the organoids, we see them quite rapidly. What we currently don't know is that what we see in vitro coming back, so the recent ones, whether they are predictive of what would happen in the patient. Doesn't mean that's of course a long-term study, but it would be really nice if you could actually first find a drug that kills off 99% of the cells, but then already know, okay, but then there will be escapers, but we already know who they are, and we have another drug that will kill those. That is currently not known, but that would mean if you think about this study, it's quite complicated, but that would be very interesting. Yeah.
I just have a question, a curiosity. You showed this beautiful video where there are some CAR T cell which seem to be completely unable to kill tumor cell and this serial killer. So we have recently published together with others that immune cell can undergo to cellular senescence. So have you looked into that? And particularly, I'm interested to know if perhaps this anergic T cell, which do not do anything, also can antagonize the effect of the killer somehow. So have you tried to remove it to keep only the killers to see if you improve the anti-tumor?
I don't think that has been addressed in that study. What actually is addressed in that study is also quite unique observation, is that what often happens before these cells start killing, that another T cell goes into an organoid, doesn't kill, but touches the organoid, comes back out, touches one of these cells, and from then on, this cell is the serial killer. We have no clue what the signals are, but it might be that this has to do with awakening cells from senescence.
Thank you. Thank you very much for the, sorry, we don't have to. Let me introduce. So let me introduce René from here. So I can come here. Okay. So it's a great pleasure to introduce the next speaker, which is René Bernard. René is a PhD, is a training, a very famous scientist, and also a very good friend of mine. We have the pleasure to work together for many, many years in the field of cellular senescence. So René is a professor of molecular carcinogenesis at the Netherlands Cancer Institute, and is the current president of the European Association for Cancer Research. So today we are very lucky. We have two presidents, one president, one CEO of two different associations. So it will take very long to summarize all the CV, but I have tried to do this by listing the three most important contributions of René to the field of cancer biology. So the first one that probably people do not know is that René has been the first scientist to clone and discover the function of RB genes, which doesn't mean retinoblastoma gene, but it's René Bernard gene. So it's been named by René, right? So, and when he told me, I was really astonished. Right? So the second is that René has developed the FDA-approved combination of a BRAF inhibitor and a EGFR inhibitor for the treatment of BRAF mutant colon cancer that has saved many, many lives. And finally, that he has also developed for the first time a clinical use gene expression test for early breast cancer prognosis, which is called MammaPrint, and that he has founded a company, very successful, it's called Agendia, where he has spent many, many years to bring to the clinic this important tool. So the René Lab is well known for the use of functional genomic approach to find the vulnerability of cancer that can be exploited therapeutically. The title of his talk is Unconventional Approach to Cancer Therapy. Thank you, René. The stage is yours. Yeah.
So thank you, Andrea. The question that I want to address with you is whether we can learn something from using 2D cell models. I hope that at the end of the day, you'll agree with me that the answer is potentially yes. The work that I'm going to talk about is related to a company that I interact with called Lixti Biotechnology Holdings, and you'll see the drug that they develop has some potentially interesting uses in the clinic. So we're all excited about targeted therapies. They do better than chemotherapies, but of course, in the end, patients progress, and that's a problem, right? So in the end, we see that the Kaplan-Meier curves of the chemotherapy and the targeted therapy really converge on a very low probability of survival. So we know already what the answer is to this type of problem, right? Single agent therapies don't solve the problem, combinations do. That was the answer for AIDS. It's probably also the answer for cancer. This is not new, because if you look at the first paper on combination therapies, it dates back to 1958. So this whole idea that combinations are the answer to this problem of cancer drug resistance is not a new idea. The only problem that we're having today is that we have so many drugs that you can make nearly endless number of combinations. So we need to devise smart technologies in the lab to find the best combinations, because you'll probably agree with me that we cannot test all 499,000 two-way combinations for every cancer type. That is not a viable concept. So how do we reduce this number of possible combinations to a manageable number, and how do we approach this in the lab in vitro? So I'm going to talk a little bit about synthetic lethality to improve drug response. Synthetic lethality is, of course, a genetic term that was originally used to show that two genes that are individually non-lethal become lethal in combination. I'm now going to apply this concept to drug synthetic lethality. That means two drugs that are individually non-lethal become highly lethal in combination. How do we find these lethal pairs of drugs? And Andrea already introduced this, but I'll just give this as an example, as an introduction, and then go into very new territory with you, how we exploit this concept of synthetic lethality. So back in 2004 or so, we began to see the first responses to targeted agents with EGFR inhibitors, and shortly thereafter, we began to see that BRAF inhibitors worked really well, at least initially, for BRAF mutant melanomas, even though resistance there, of course, also occurs after single agent therapy. But the big surprise, of course, was at the time that we thought if you know the mutation, you know which drug to use, because people thought the melanomas with the BRAF mutation are addicted to the BRAF signal, and if you take that signal away, they die. The concept of oncogene addiction, coined by Bernie Weinstein, was thought to be generally useful, and therefore we thought we just need to sequence every patient, and then we know how to treat the patient. Then came a rather dramatic turn of events when Scott Kopetz thought he also then had the answer for the BRAF mutant colorectal cancers, because the mutation in the colorectal cancer is exactly that you see in the melanoma, V600E in the BRAF gene. So he thought, if I use the very same drug that is so successful in melanoma, I'll get similar responses in the colorectal cancer patients. And the answer was he didn't. He saw nearly no responses. So this was a blow to this whole concept of if you sequence, you'll know what to do, because it turned out that the context in which that mutation occurs is also important. So this was a disappointment to many, but I saw this as an opportunity, because we had just developed in the lab this technology to silence genes at will. We had developed this plasmid. This was in the year 2011 BC, remember? BC stands for before CRISPR. We had short hairpin RNAs that we could use to silence genes, and we had just developed the first library that covered all the kinases, a hairpin library that inhibited all the kinases. And what we did here is that we took a BRAF mutant colorectal cancer cell, did not respond to this BRAF inhibitor that was so effective in melanoma, infected this with this short hairpin RNA library, such that one kinase was inactivated in each colorectal cancer cell, and then split the population in half. Half of treated was regular culture medium, and the other half with this BRAF inhibitor, vemurafenib. Now remember, vemurafenib does not affect the proliferation of the colorectal cancer cells. So now we cultured these cells for about 2 weeks, and then we gave each of these hairpin vectors a different barcode, and I give you the barcodes a separate color. And the question was, is there any kinase whose inhibition becomes lethal not by itself, but only in combination with vemurafenib? That would be a synthetic lethality with vemurafenib. And the way we can identify such a synthetic lethal interaction is that we recover these barcodes by PCR and simply sequence them by deep sequencing and then quantify them. And if there's one barcode that is missing only in the presence of vemurafenib, then the kinase encoded by that blue barcode is synthetic lethal with the BRAF inhibitor, right? So that was the simple experiment. Even though it looks simple, it was at the time technically quite challenging. But the answer that we got out was that the blue barcode encoded a hairpin for the epidermal growth factor receptor, EGFR. Now again, EGFR is not at all lethal in the BRAF mutant colorectal cancer cells. The BRAF inhibitor is not lethal, but the combination of the two became highly lethal, as you see here. So this is an example of two drugs that are individually non-lethal that become highly lethal in combination. And we call this drug-drug synthetic lethality. And of course, no clinician in his or her right mind would test two drugs that have no single agent effect and test those in a combination. But this experiment suggested that this was a combination that was potentially worth testing. So we did this, of course, first in an animal model, where you see again that the single agents don't work, the combination did work. And that was a publication that was from 2012. And since I work in a comprehensive cancer center where we have very low threshold interactions with our clinicians, we were able to recruit the first patient within 8 months after the publication in a proof of concept trial where we combined cetuximab as an EGFR inhibitor with encorafenib as a BRAF inhibitor. We saw the initial responses in that publication. We then went on to do a large phase 3 global study that showed the efficacy of the combination of oncorafenib and cetuximab. That was published in 2019, and that resulted in this FDA approval in 2020. So here you see the timeline from experimental design to an FDA approval. Some people say 8 years is fast. I think it's unacceptably slow, and I hope that Hans will do something at Roche to make sure that these timelines will be shortened in the future so that our discoveries can make it faster to the clinic than they currently do. So I want to now go back and talk about another problem, and that is the problem that we thought we can now solve every problem in the clinic, namely that single-agent MEK inhibitor is not very effective in KRAS mutant tumors. You would expect that that would be the case, right? RAS is upstream of a kinase cascade, RAF, MEK, ERK, so we thought that if you now inhibit MEK downstream of KRAS, you should get a clinical response. Well, again, the surprise was that that was not the case. Single-agent selumetinib did not work for KRAS mutant tumors. So we thought, we know what to do. We'll do another of these synthetic lethality screens. And we did. And what we found is that MEK inhibition in KRAS mutant tumors leads to a reactivation of now another receptor tyrosine kinase called ERB3, and that ERB3 activation leads to an increased signaling through the MAP kinase pathway such that the MEK inhibitor is inefficient. So we thought the answer is, again, a simple combination of, in this case, a pan-HER inhibitor, because ERB3 can form heterodimers with EGFR and HER2. So you need to inhibit all members of this HER2 family. And there's a number of drugs that do that. One of them is called dacometinib. And you saw again that in our cell models, selumetinib and dacometinib was very good. And in our animal models, it was very good. We got very complete tumor control with dacometinib, or in this case, afatinib and trametinib, a MEK and a pan-HER inhibitor. So we were very excited. We thought, another one that we can cure. And this is a much bigger one because there's so many KRAS mutant tumors. And now comes the kicker. We tried 3 different combinations of either afatinib, dacometinib, or lapatinib in combinations with different MEK inhibitors in the clinic. And what was the answer? The answer was that this drug combination of a pan-HER and a MEK inhibitor is so toxic that you never reach an effective concentration of the 2 drugs to inhibit the MAP kinase pathway, because you see toxicity long before you see efficacy. Right? We didn't see that in the mice. The mice were perfectly happy with a high concentration of the 2 drugs. So here you see an example where the mouse is completely non-predictive for human toxicity, right? And so this is also a warning. Don't, overestimate or don't overinterpret what you learn in your mice, because in patients, the situation may be very different. So knowing that an RTK inhibitor and a MEK or ERK inhibitor was not a good idea, we started to think about other approaches. And here you see that, in fact, the MAP kinase pathway is also dependent on a connector molecule, a phosphatase, called SHP2, that connects receptor tyrosine kinase to RAS, MEK, and ERK signaling. And conventional wisdom had it that if you have a mutant RAS protein, that in fact that mutant RAS protein is independent of upstream signals, so that the upstream signal is no longer required for RAS to transmit its signal through the downstream pathway. So that would suggest that if you actually now, let's see, how do we do this? Here, remove this protein SHP2, that potentially the cells would stop growing, would not care about that, because the signal would not be required from upstream. And in 2015, we actually did that experiment. We knocked out the gene encoding SHP2 called PTPN11, and we saw that if you knock out PTPN11 in a human lung cancer cell that is KRAS mutant, the cells are unable to form tumors. In fact, they show a senescence response, which was very surprising because the cells were perfectly happy to grow in cell culture in 2D. But when you injected them in vivo, they stopped growing. And we saw the same for the small molecule SHP2 inhibitor, SHP099. Again, signs of senescence response, senescence-associated beta-galactosidase, and no proliferation in vivo. So that resulted in this notion that potentially you could also interfere with RAS by upstream inhibition with a molecule, small molecule inhibitor of this phosphatase called SHP2. And we thought that potentially this would be very useful, especially when you combine it with a downstream inhibitor of RAS. So upstream and downstream dual inhibition could be potentially helpful. And Sarah Mainardi, who spearheaded this work in my lab, showed that if indeed you take the inhibitor called RMC-4550, which is a SHP2 inhibitor, as a monotherapy, the tumors are somewhat inhibited, but not dramatically. This is change from baseline growth from mice. So normal mice that are treated with vehicle, the tumor grows. There's a little growth with the SHP2 inhibitor. The ERK inhibitor as a monotherapy has very little effect, but the combination of a SHP2 and an ERK inhibitor, dual upstream-downstream inhibition, leads to tumor regressions. And this experiment formed the basis of a trial that is funded by the Stand Up to Cancer AACR Lustgarten Foundation Pancreatic Challenge, a trial where we combine a SHP and an ERK inhibitor for KRAS mutant tumors, and this is now in the 3rd dose escalation levels, and we're beginning to see some interesting things happening here in the clinic. So this is an ongoing project. I now want to turn to something that will probably make you think that René is now getting old and he has lost it, and he is now, maybe it's time for him to retire and stop working on cancer, because what he's doing now is a bit crazy. Some old men start to do crazy things. We have a few old scientists among us that start to say crazy things. So I would be in good company if I did, but I hope that you will agree with me after this few slides that what I'm going to say to you may not be as crazy as your first reaction probably will be. So this is the work of Matheus Henrique Dias in the lab, and it's a special form of synthetic lethality that is based on a deliberate hyperactivation of oncogenic signaling in cancer cells to accomplish cancer cell death. So why would we do that? So think about the famous hallmarks of cancer, right? We try to, what cancer cells do through oncogenic signaling is in fact that they acquire some of these properties that we know from the hallmarks of cancer, which is insensitivity to anti-proliferative signal, limitless proliferative capacity. But what you see in these hallmarks of cancer, if I can get my arrow to work, which is probably not going to work, that on the bottom side, you see that these cells also activate all these stress responses. Stress responses like DNA damage stress, oxidative stress, mitotic stress, proteotoxic stress. This is inherently linked to the fact that cancer cells are hyperactivated in their mitogenic signaling. So this is the price that cancer cells have to pay for being cancer cells. They have to live with stress responses. Nearly every cancer cell that you stain for gamma H2AX is positive because it has DNA replication stress, right? So there's an inherent message in this, namely that cancer cells probably do not strive for the maximal activation of oncogenic signaling, because there is a level of oncogenic signaling where the stress responses become so dominant that they would become inhibitive for proliferation. So the notion would be that tumorigenesis selects for optimal oncogenic signaling, that leads to optimal proliferation. It is not striving for maximal oncogenic signaling, because otherwise the stress responses would become dominant and would become prohibitive. So in conventional therapy, we try to inhibit the oncogenic signals, and we all know what happens when you do that. If you inhibit oncogenic signaling, the cancer cells manage to get a secondary mutation that reactivates the oncogenic signal, and then you're lost because the cancer cell proliferates in the presence of the drug. So the alternative that I'm proposing to you today is to not try to inhibit oncogenic signaling, but rather to super activate the oncogenic signaling, because yes, that would potentially stimulate the cells to grow faster, but it would also hyperactivate their stress responses. And potentially, if you did a combination, and this is why I call this a special form of synthetic lethality, it's not about only activating the oncogenic signaling further, but you should do so in combination with a perturbation of the stress responses that the cancer cells need to survive in the presence of these hyperactivated oncogenic signaling. And this combination of superactivation of signaling and perturbation of the stress responses we call now a paradoxical intervention, because it's completely counterintuitive. You're doing what you actually think you should not be doing, right? So does this make any sense, and is there any way to make this done? Now think about this paper that no one less than Harold Farmers published in 2015. He had a mouse strain that had a mutant EGFR in the lung epithelium. That mouse developed lung cancer. He had a second mouse that had a KRAS oncogene in the lung epithelium. That mouse also developed lung cancer. But when he crossed the two mice, where the lung epithelium now had two oncogenes, KRAS and EGFR, there was no more lung cancer. And that shows that too much oncogenic signaling is incompatible with cancerous growth. So here you see the example that, in fact, too much of a good thing is not a good thing for a cancer cell. So how are we going to activate oncogenic signaling further in a cancer cell that already has a high level of oncogenic signaling through the mutations that cause the cancer to begin with? Check out protein phosphatase type 2A. It's an inhibitor of MEK signaling and ERK. It inhibits Wnt beta-catenin signaling, and it inhibits PI3 kinase AKT signaling. So in fact, 3 of the major signaling pathways that we know to be deregulated in cancer are inhibited by PP2A. So what would now happen if you inhibit PP2A with a drug? That should lead to a further activation of MEK-ERK signaling, a further activation of Wnt beta-catenin signaling, and a further activation of PI3 kinase AKT signaling. Fortunately, there is such a molecule. It's called LB100. It has been developed clinically in combination with chemo and radiotherapy. And surprisingly enough, in the phase 1 trial, it showed a very modest toxicity, where the grade 2 fatigue was the highest level of toxicity seen, which is really quite modest. This was very unexpected, I may say, because there is one drug that everybody uses in the lab experimentally to inhibit PP2A called okadaic acid, and it's toxic like hell. But okadaic acid is structurally unrelated to PP2A, and it turned out that the toxicity of okadaic acid is probably unrelated to the inhibition of PP2A, but is an off-target effect, because this molecule, LB100, has no structural resemblance and has no toxicity in mice or in patients. So if we treat colorectal cancer cells with LB100, you see that there is an anti-proliferative effect. Anti-proliferative in the presence of hyperactivation of mitogenic signaling. Here are 2 colorectal cancer cell lines. HC29 is BRAF mutant, SW480 is KRAS mutant, so they have both got activations of the MAP kinase pathway, but you see at the top line that the level of phosphor-ERK is dramatically increased by this drug LB100. So you also see that phospho-Jun is activated, and that the increase in targets of Wnt beta-catenin signaling like axin-2 are increased. You also see that the stress responses are activated as a result of that. We see gamma H2AX, phospho-CHEK1, unfolded protein response, phospho-IRE1 alpha, and that probably explains why these cells have, on the one hand, more oncogenic signaling. They actually do grow slower because of these stress responses that are activated. So we have a drug that can superactivate mitogenic signaling, and it leads to proliferation inhibition rather than further stimulation because of these stress responses that are activated. But I told you this is probably not enough. We need to somehow perturb these stress responses, right? So the question is, which of the stress responses do we need to perturb to kill these cancer cells most effectively? So what Matthias did, he made what he called a stress-focused drug library, where he found 164 drugs that either affect metabolic stress, proteotoxic stress, mitotic stress, oxidative stress, DNA damage stress, and tested all of these drugs in 15 concentrations in the presence and absence of this drug LB100. And he asked a simple question: is there any drug that works better in the presence of LB100 than by itself? And here you see the sort of answer for that experiment. These are plots where we measure the area under the curve of the drug plus minus LB100. The further down a dot is, the more toxic the drug is in the presence of LB100. And you see in two different cell lines, we see inhibitors of WEE1 and inhibitors of CHEK1 stand out. So WEE1 or CHEK1 inhibition appears to be particularly toxic in the presence of this drug LB100. So knowing that that was potentially interesting, Matthias asked, let's now do an unbiased selection. Let's do a CRISPR genome scale to see which gene is the most toxic when we treat cells with LB100. And the answer was, again, that WEE1, that we also found in the compound screen through the drug adavosertib, the WEE1 inhibitor, WEE1 was highly toxic in the presence of LB100. Since WEE1 is in clinical, inhibitors are in clinical development, and we felt that that was the one that we should follow up on. So here you see a whole panel of colorectal cancer cell lines where we asked the question, is there synergy between the LB100 PP2A inhibitor and the WEE1 inhibitor adavosertib? And you see that in each case, there is a dramatic synergy independent of whether it's a BRAF mutant, a KRAS mutant, or a double wild type cell. In all the colorectal cancer cells, we see a striking synergy between these two drugs. So how does this actually is not only true in colorectal, it's also true in pancreatic cancer cells and in very untreatable form of cancer called cholangiocarcinoma. So this is probably something that we can use quite broadly. So how does this work? Why is a WEE1 inhibitor synthetic lethal with a drug that activates mitogenic signaling dramatically? And the answer is probably that LB100, by superactivating mitogenic signaling, creates DNA replication stress. It forces cells through the cell cycle by hyperactivating the mitogenic signals. And the WEE1 inhibitor is a drug, WEE1 is a kinase that prevents cells to enter mitosis prematurely before DNA replication is complete. So now you can imagine that if you push cells through the cell cycle and lower the threshold to enter into mitosis, what's going to happen? Cells with unreplicated DNA are going to enter mitosis, and you know what happens then. Then you get what we call mitotic catastrophe. And that's exactly when we do time-lapse filming of cells that are either treated with LB100 monotherapy, adavosertib monotherapy, the WEE1 inhibitor, or the combination. Cells treated with the combination enter mitosis, but never come out again. They die in the process. And if we look at the chromatin of cells that are actually in the combination treatment, the chromosomes, they are no longer recognizable as individual chromosomes. We call them pulverized chromatin. The chromosomes are a complete mess, and that's why the cells die in mitosis from apoptotic cell death. So the cells have been pushed into mitosis prematurely by the hyperactivated oncogenic signaling, and as a result of that, they are now dying by mitotic catastrophe. We can show you that these cells actually do exactly that, that cells that have between 2N and 4N DNA content, and these four are cells that are in S phase, these cells already stain positive for the mitotic marker is phosphohistone H3, and that means that these cells are in mitosis with DNA content between 2N and 4N, and they should never be in mitosis with a halfway replicated DNA, and that's why they crash in mitosis, because you see here that the phospho H3 positive cells in the combination treated cells is rather striking. So we think we understand how it works, and we think that this is also why the inhibitor WEE1 is synergistic with chemotherapy. Chemotherapy also gives you replication stress, and WEE1 then allows the cells to enter mitosis in the presence of replication stress. So the question can be asked, are we now just doing a phenocopy of this simple experiment, cause replication stress, kill the cells in mitosis, right? And there was recently a positive study, azacitidine plus chemotherapy in ovarian cancer, that was positive. In spite of the positive phase 2 trial, the next decision that AstraZeneca took was to discontinue clinical development of adavosertib. The logic of that is that they had spent so much time to figure out which combination was the best one that they said by the time we have a phase 3 that is active, our patent life is too short and we don't care anymore. So fortunately, there's another company that has a WEE1 inhibitor called Zentalis. And so we think that the WEE1 inhibitor will be continued to develop. But what I'm going to show you here very quickly is that, in fact, the combination adavosertib plus doxorubicin or gemcitabine that is active, and there is synergy, that the synergy of LB100 with adavosertib is a much greater synergy than that we see with LB100 and chemotherapy or adavosertib and chemotherapy. So we believe that the combination that we have developed here of the LB100 plus the WEE1 inhibitor is superior to the ones that are now in clinical development of chemotherapy plus WEE1 or chemotherapy plus LB100. If you go to experimental animals, these are orthotopic models of colorectal cancer transplanted into the colon of mice. We see that the combination is dramatically effective. The one thing that I think you want to notice is that in the lower panel where we treat with adavosertib and LB100, there is some tumor volume left, but it's mostly reactive stroma and dead cells. There's only a small rims of viable tumor cells left that are responsible for a very small fraction of the tumor volume that is remaining in these animals. So we think that this combination is very potentially effective and also has a very modest, if I can get the next slide too. Yes, there it was. In the absence of any detectable toxicity that we see in these animals. I said this works also for other models. Here you see a cholangiocarcinoma model. Cholangiocarcinomas have a terrible prognosis, no agents that work against them other than chemotherapy. And you see here also a conventional PDX xenografted model of LB100 with adavosertib, leading to significant responses in the absence of weight loss. So now the part that I think is the most exciting. How would you, if you were a cancer cell, develop resistance to this type of therapy? We all know from the last 20 years of work that if you inhibit oncogenic signaling, the cancer cell tries to reactivate it. So my simple-minded view was that if cancer cells are faced with a hyperactivation of oncogenic signaling, and that is the stress that they're in, they will try to reduce the oncogenic signaling. Now, wouldn't that be nice if you could convince cancer cells to reduce their own level of oncogenic signaling, because if we then take the drug away, there's no oncogenic signal left. So can we force the cells with this type of approach to evolve towards a less malignant phenotype? Is that the resistance that will develop? So the first thing we did is ask in a genome-wide CRISPR screen which genes, when we overexpress them, make the cells more sensitive to LB100. This is what we call a CRISPR-A screen, where we overexpress genes to ask when do we make make the cells more sensitive to LB100. And there you see the top outlier MYC, you see MYP, you see cyclin E, you see cyclin D1. These are all oncogenes. If you make oncogenes more active, the cells become more vulnerable to this drug. Conversely, if you do a CRISPR knockout screen and you ask which genes are positively selected in the presence of LB100, so that means how do we get resistance to this drug, then we see again oncogenes. Why? If we knock out beta-catenin as a top hit, we reduce Wnt signaling, and now the cells are resistant because they have now reduced their oncogenic signaling in order to develop resistance. And if we put all these genes in a string network, then we see that there's two nodes, one node around MYC and one node around beta-catenin, that say that MYC and beta-catenin signaling are genes that control the response to this drug LB100, consistent with the notion that LB100's toxicity relates to changes in MYC and beta-catenin signaling, right? So, when I saw this result, I had this idea that dates back 40 years when I was young and a blonde Adonis, as March knows. At least I was described as such in a book at the time. It wasn't true, by the way, but then again. I did an experiment in the lab of Bob Weinberg. I overexpressed NMYC in neuroblastoma cells with the aim to understand why NMYC-amplified neuroblastomas are so aggressive. And one thing that we learned then is that NMYC amplification leads to downregulation of class 1 HLA antigens, that leads to immune escape of the cells. Nobody at the time cared because nobody thought that, in fact, the immune system had anything to do with control of cancer growth. Little did we know then. But I still had these cell lines, and I simply used now these cell lines again to show that when NMYC is increased in expression, we made transfectants of NMYC in neuroblastoma cells, that now these cells become more vulnerable to LB100, and that means that more MYC makes you more vulnerable to LB100. So how do cells then now spontaneously, right? Now I can show you how I can modulate experimentally the resistance. But if I give cancer cells the choice, you figure out how you want to develop resistance to adavosertib plus LB100. Your call, what do you do? Well, if you grow them for 4 months in the combination drug, it does work. We do get resistant cells, right? So how do these resistant cells now look? The first and probably most striking effect is that cancer cells typically are highly aneuploid. And the SW480 cells that we made resistant have, on average, some 80 to 90 chromosomes, right? Aneuploid cells are highly oncogenic. The resistant cells have reduced their karyotype to near diploid, 50 chromosomes. So here you see already an example where the evolution is to a normal phenotype, less aneuploidy as a result of drug resistance. If we go to single cell sequencing of the cells, we see that the resistant cells are very different from the parental cells. And if you look at the pathways that are most affected, we see that MYC targets are down in the resistant cells, E2F targets are down in the resistant cells, and beta-catenin targets are down in the resistant cells. 3 pathways that we know that contribute to oncogenic signaling and to malignant growth are dramatically downregulated as a result of resistance development, right? And that is also seen in the Western blots, where we see that the mitogenic signaling is down, but also those stress responses are, of course, now down. There's less gamma H2AX, there's less phospho-CHEK1 in the resistant cells, because if you downregulate oncogenic signaling, you have less problems with your stress responses, right? And now, I want to show you the most, I think, dramatic result of the entire set of experiments. When we grow these cells in 2D, the resistant cells can grow. If we grow them in 3D, the resistant cells are simply unable to grow. Now that's a surrogate for tumorigenicity, but the proof of the pudding is in the injection of these cells in mice. And here you see the result of that experiment. SW480 cells are highly tumorigenic and form tumors in nude mice, but the cells that had in vitro developed resistance to LB100 plus adavosertib are unable to form tumors. They have lost their aneuploid phenotype, they have lost MYC, they have lost Wnt, they have lost E2F, and they have lost their oncogenic phenotype. So I think that hyperactivation of oncogenic signaling selects for resistance that escapes or that downregulates the oncogenic phenotype, and as a result, we get a sort of tumor suppressive resistance. We call this a paradoxical intervention because my wife is in psychotherapy, and she tells me that in psychotherapy, a paradoxical intervention is characterized by a deliberate reinforcement of the pathological behavior. You would have to agree with me that that's what we do when we hyperactivate oncogenic signaling. We deliberately reinforce the pathological behavior to improve the clinical condition. Now, if you get resistance to this therapy that leads to loss of oncogenic phenotype, potentially we can improve the clinical condition. And I think we would also agree that this counterintuitive approach should only be considered when more conventional therapies failed. So after resistance to targeted therapy, we want to try this. And I hope that by the end of this year, we'll have the first patient on trial where the WEE1 inhibitor and LB100 are combined in patients that fail to respond to conventional therapies. So my take-home message is that hyperactivation of oncogenic signaling is, in my view, a viable cancer treatment strategy because resistance selects for cells with reduced oncogenic phenotype. And I think the overall message is something that I also wrote in Cell a few weeks ago. It's combination stupid. The answer is in principle simple, but we need to spend a lot more time as a scientific community to find the right combination of existing drugs. It is not really in the interest of large pharmaceutical companies to find effective combinations of existing drugs, because of course existing drugs sooner or later run out of patent, and then the business model of big pharma is lost. So that's why there's a continuous source of new drugs, but we all know that new drugs have the same problems as old drugs. The G12C inhibitors, everybody was super excited about G12C inhibitors. The Lancet in March published that long-term survival to sotorasib is as good as doxorubicin, right? There's no better improvement than that. So we need combinations over time. And if we would use more combinations of existing drugs, of course, over time, this would really contribute to affordable healthcare because it would be more combinations of existing and therefore generic drugs that could potentially be very beneficial to patients. So on that note, I'll stop. The SHIP-ERG story was done by Sarah and Antonio and Astrid, and the paradoxical intervention nearly exclusively by Matthias with help from the lab of René Medema and Alberto Villanueva. And I'll stop there and be happy to answer any questions.
Thank you. Thank you, René. I want to go first because I was amazed by this last part of your seminar, right? And we have done something very similar for a long time in the lab. So when I was a postdoctoral fellow, I had a project where I was hyperactivating the PI3K pathway through PTEN inhibition.
Yeah.
So I don't know, there is a drug, it's called Vopic, that has been used also clinically later on. And I think you're perfectly right. So this is an amazing approach. So we didn't have the idea to do the double combination at that time. But my question for you is the following, right? So are you afraid, so hyperactivating pathway will also elicit cellular senescence. So you will not have like an hyperproliferation in certain conditions. So you may have senescence and perhaps you will not be able to drive a mitotic catastrophe to a second drug, right?
Yeah.
The other question, if you even expand your paradigm, so you try to combine different activator of pathway, so like PI3K activators and MAP kinase activator. What is going to happen in your opinion?
Well, we're going to try that. Obviously, there's drugs that did very well in the clinic. A drug called prostratin that was developed for the treatments of AIDS, which is a PKC activator. There's of course lithium that has been used in the clinic for decades to treat bipolar disorder, which is a known inhibitor of GSK3 beta and therefore leads to Wnt activation. So you would think intuitively that if you activate Wnt, that many initiated cells will in fact start to grow faster and that you would in fact promote tumorigenesis by treating patients with lithium. There have been epidemiological studies in patients treated for bipolar disorder with lithium, and the answer is these people get less cancer rather than more cancer. So in fact, activation of Wnt signaling in patients is also anti-tumorigenic and not promoting growth. And those of you that study the Wnt field closely, Louis Vermeulen had a beautiful paper in 2021 in Nature where he had the APC min mouse where he activates the APC in activation, gets all these adenomas in the colon, and then, or in the small intestine, I should say, and then he pre-treats with lithium chloride and then sees that in fact he gets fewer adenomas. And now there is actually a trial in the Netherlands where we treat FAP patients that are predisposed to adenoma formation with low doses of lithium chronically to see whether that leads to less adenoma. So I don't think that activation of mitogenic signaling is necessarily highly toxic to normal individuals, but we'll need to see.
In diabetes, they actually use this approach, right? So there are many drugs done for diabetes, like Vopic, whatever,
Yeah.
that use this strategy to activate the pathway.
and-
so you could use it like a multiply.
And as I said, the LB100 that you would think would activate so many different signaling pathways never goes beyond grade 2 toxicity in the clinic. So it combines well.
Very interesting. There are questions. Sorry, I wasn't. Yes, a question over there.
Just to extend the last paradigm, would you achieve the same effect if you subjected the cells to hyperoxic or hyperbaric conditions? Would you stimulate oncogenic signaling, and would that increase sensitivity to your cancer drugs?
Hyperoxia, or
no,
hyperbaric oxygen.
Yeah.
I'm not sure whether that would be necessarily a mitogenic signaling.
You could oncogenic?
Because ROS is mitogenic, so in that sense, potentially higher ROS levels could lead to a higher mitogenic signal. It would be a very indirect way to do it. I've seen some results from people that treat KRAS mutant tumors with basic FGF to further stimulate signaling. And in fact, there you also see an anti-proliferative response to growth factor signaling, to further increase growth factor signaling. So if you go through the literature, and we've written this piece in Molecular Oncology in 2021 about paradoxical activation, we summarized all the literature to say that hyperactivation of oncogenic signaling is in fact reducing proliferation rather than increasing it. There's actually quite a bit of evidence in the literature if you look carefully. If you take BRAF mutant melanomas that acquire a KRAS mutation to develop resistance, you take the drug away, you get oncogene-induced senescence as well. So KRAS plus BRAF is too much signal, and there's a senescence response. Just like you said, the PTEN loss, right, is also senescence response.
There is another question here.
Thank you. I think that one of the main problems is the fact that we have wonderful results in basic research in vitro, in animal studies, but when we are going to clinical trials, we see already that doesn't work so well, doesn't work. The main problem I may imagine is, all you know, is the fact that we are treating very advanced patients. We start treating very advanced patients. Mm-hmm. We don't see the results so well. So one of the points that, for example, we try something very similar to what you just explained now. We know that the MDM2 amplification, it's deregulate the P53, and by inhibiting the MDM2, we thought that we may have good results in clinic having a normal P53. But it was wonderful. We got wonderful data in basic research and preclinical studies, but it didn't work in clinics. So the idea maybe should be, again, to work in adjuvant setting more than in metastatic disease and advanced disease.
So I showed you one combination that worked very well, the BRAF EGFR, that is now FDA approved. I showed you the combination of MEK plus PANHER that was a disaster in the clinic, both of which were very rational from a basis of understanding signaling. So yes, sometimes it works and sometimes it doesn't, and it's hard to predict what will work in the clinic. What I will say to your question of in vitro versus in vivo, the PDX models that we used with Alberto Villanueva, these are derived from patients that have resisted 4 or 5 lines of prior therapy, right? These patients don't respond to anything anymore. At that point, they go into mice and we try this combination. So in that sense, it resembles a 4th or 5th line of therapy, and we did get meaningful responses in that setting. So that gives me some reassurance that if we go to 3rd line therapy with this in the fall in KRAS mutant colorectal cancers, which is our plan, that we may be able to see something. But we'll see. There's no guarantees, but I still believe that these combinations that we develop that have a solid and rational basis are far more likely to be effective in the clinic than what I call the fool around and hope to get lucky combinations that you often see in the clinic today. 11,000 trials combining PD-1 or PD-L1 with something else. You can't make me believe that there is any rationality in each and every one of those 11,000 trials, right? This is just hoping to find a signal, right? We are not hoping to find a signal. We first come up with a rational combination, we fully understand how it works, and then we go into the clinic. And that is, I think, acceptable to patients, that there is a solid underpinning of why we think that combination should work. And it's by far not true for the vast majority of clinical trials that are ongoing today.
Thank you. Well, just last question, Miles, and after I have to...
Very much for your talk. I really enjoyed, especially this type of, paradoxical type of research. I think is quite fascinating. And I'm in the longevity medicine kind of side of things, and a term they throw around a lot is hormesis. I mean, is that is this pharmacological paradigm kind of the lens through which you view some of your research as well?
Hormesis, you say?
Sorry, hormesis.
Hormesis. And that means?
Hormesis refers to biphasic dose responses in a number of different life science. We can all discuss this later if it's, but...
It doesn't ring a bell immediately, but as I said at the beginning, I think there is a Goldilocks scenario for oncogenic cycling for cancer cells. If you go below, they won't grow. If you go over, they won't grow. So the cancer cells need to balance it very carefully. And that's why I think we have this window on either end, right? You can go over or under and they will respond.
No, I mean, it's super cool. And I think about a lot of the kind of longevity medicine scientists where they talk about caloric restriction or inducing low levels of cellular stress to extend lifespan as being hormetins, and it's almost like you've jumped to the other side of the bell-shaped dose-response curve, and it's cool. Thank you.
I think the confounder in that caloric restriction is that people say you live longer, but maybe it is only that it seems that you live longer because you have such a horrible time being hungry all the time. I don't know. Anyway, I'll stop there.
So last but not least, it's my pleasure to introduce Arianna Baggiolini, which is a very young and talented scientist and assistant professor at the University of Svizzera Italiana. It's also a group leader in our institute, at the Institute of Oncology Research. Arianna just came from Memorial Sloan Kettering and just opened the lab a few months ago. Arianna studied melanoma, which is the most aggressive skin cancer. She's interested in defining how developmental programs are reactivating during the acquisition of malignant state, and how this can lead to tumor formation and progression. So the title of her talk is Patient-Specific Models of Multicellular Oncogenic Competence in Metastasis. Arianna, the stage is yours.
Hello, everybody. It's just terrific to talk after Hans and René, and I will try my best to keep the excitement high in the room. Now, we are aware that the majority of our healthy tissue accumulates a tremendous number of mutations over time, and this has been greatly elucidated by the seminal work of Peter Campbell and colleagues. Now, let's have a look for a moment at what happens to the healthy skin. And here you have a schematic representation of the number of clones and the size of those clones that carry cancer-causing mutation and that can be found in 1 square centimeter of healthy human skin. Now, luckily for us, we know that only a minority of these mutations will eventually result in malignant transformation and tumor formation, really highlighting the fact that oncogenic mutations are, yes, necessary, but not sufficient for tumor formation. And this is really prompting the question of what defines the potential of an oncogenic mutation to induce malignant transformation. What defines oncogenic competence? And with oncogenic competence, I'm referring to the fact that most DNA mutations are tumorigenic only in certain cellular contexts. Now, let's imagine again for a second in our skin what happens to a melanocyte when it acquires an oncogenic BRAF mutation. Well, a new oncogenic mutation is comparable to a new input of information, and this new input of information can be differentially read depending on a pre-existing cellular black box. And the result can be tremendously different. You can have either malignant transformation and melanoma formation, or benign transformation and the formation of a benign nevus. Now, what is this pre-existing cellular black box? This is everything that defines specific cells. Those are all the epigenetic and transcriptional program of a specific cell, and we know nowadays that those programs are lineage specific, they are clearly differentiation state specific, and they are highly regulated by the microenvironment as well. Now, to try to define how the differentiation state of a cell inside a specific lineage trajectory regulate oncogenic competence, we develop a human pluripotent stem cell-based cancer model. Human pluripotent stem cells have the potential to give rise to any cell types in our body. They can be either from embryonic origin or they can be patient-derived upon reprogramming of adult tissue like fibroblasts or peripheral blood like T cells. And what we did was to develop a protocol to differentiate human pluripotent stem cells towards the melanocytic lineage, and this was allowing us to isolate along the way all the different stages of development along the melanocytic lineage, which include the multipotent nual crest stage and the progenitor state melanoblast. Now, at this point, we genetically engineered those cells to be triple knockout for 3 tumor suppressors that are frequently lost in melanoma patients, and on top of it, to express a doxycycline-inducible oncogenic BRAF mutation. Now, at this point, we were having at disposition 3 different cell types that actually represent 3 different differentiation stages along the melanocytic lineage that carry exactly the same genetic background. And the simple question that we asked was, are those genetic mutations equally efficient in inducing malignant transformation and tumor formation? Well, the answer was a clear no. So what we realized was that human progenitor cells, both neural crests and melanoblasts, were readily transformed by those mutations and able to give rise to tumors in vivo, while mature melanocytes were by far less efficient, and they actually require additional mutation to be accumulated to really be able to undergo malignant transformation. And this was true in the case of human cells upon transplantation into immunocompromised animals, but also in an in vivo model, in an in vivo cancer zebrafish model, where we repeated practically the same experiment in a p53 knockout background. We were able to induce oncogenic BRAF mutation at different stages of maturation using stage-specific promoters. And again, we could demonstrate that animals that obtained the oncogenic BRAF mutation in progenitor cells were affected by aggressive tumor formation, while animals that obtained the oncogenic BRAF mutation in mature melanocytes were rather affected by apoplastic lesion. Now, to make the long story short, what we realize is that human progenitor cells are actually oncogenic competent, thanks to the expression of a number of developmental epigenetic factors that allows reprogramming and rewiring of their transcriptional state upon oncogenic expression. We showed also that mature cells like melanocytes are by far less competent. They require additional loss of tumor suppressors. However, competence can be reacquired through the action of some of those developmental epigenetic factors, and we showed as a proof of concept how it was working with eta2. Eta2 is an acetylation reader, and we demonstrated that it's actively regulating two key pathways required for malignant transformation. One is the activation of a progenitor signature, and the other one is boosting the MAP kinase pathway through action together with c-MYC. In these cases, those melanocytes are partially dedifferentiated and readily transformed by those mutations. We were excited about this data because independently on the cell of origin in melanoma, we were able to define a common mechanism of action that would allow malignant transformation, both in progenitor cell and in mature melanocyte. Now, what about microenvironment? We know very well that the microenvironment of tumor initiation is playing a fundamental role. We know equally that the microenvironment of distant organs that become distant sites of metastasis is playing a big role in regrowth of metastatic cells. And I'm going to show you in the next slides our current work, our current efforts in recreating in vitro human microenvironment of specific metastatic sites. And I'm showing in particular in the context of brain-related tumors and of primary CNS lymphoma. Now, primary CNS lymphoma is a rare subset of non-Hodgkin's lymphoma, mostly caused by diffuse large B cells, and our understanding on the human brain microenvironment and its role in tumor formation and metastatic growth is mostly relying on in vivo study, on animal models, because it's extremely challenging to study the human brain microenvironment upon isolation, for example, of post-mortem tissue cells. Those cells survive very badly after isolation. Now, animal mouse models have been and are fundamental for our understanding of the brain microenvironment of brain-related tumors. But when we are talking about the brain, we are appreciating more and more species-specific differences between human and mouse. Now, and here we are not only talking about size and morphology, but we are really talking about complexity. In fact, it has been recently shown that in the same volume of brain, the human brain shows a tenfold increase in neuronal network, and this increase in complexity is not limited to the neuronal population. It's in fact recapitulated in many different brain-specific cell types, including astrocytes, which are the neuron-supportive cells, and microglia, which are the brain-resident macrophages. Similar here, what has been observed is that human cells are larger, morphologically more complex, and most importantly, they sometimes respond even differently to some insults. And this is really prompting the need of a humanized experimental model to study the brain microenvironment of brain-related disorders. Now, you might wonder, where is actually the difference between mouse and human brain coming from, and are we able to model this difference in vitro? Well, the difference is known already from developmental biology since a while, that is occurring exactly during development already. So here you see a zoom in a mouse developing cortex at embryonic day 13, and here you see a zoom in a human fetal developing cortex at gestational week 15.5, two comparable developmental stages. Now, you can immediately appreciate that in both species we clearly have an enrichment of this neuronal progenitor population that is OX2, of an intermediate progenitor population, IOMES, around the ventricular area. However, as you can appreciate immediately, in human we have the emergence of what is called an outer subventricular area that is not present in mouse and is composed of outer radial glial cell. Outer radial glial cells are progenitor cells localized in this outer subventricular zone and are mostly found in human and non-human primates. And because of this, they are actually believed to be the evolutionary drivers of the neocortex expansion. Now, we know species-specific differences, we know where they come from. Are we able to model this in vitro? And thanks to the development of human pluripotent stem cell-based technology and the seminal work of Lancaster and Jürgen Knoblich in Vienna that generated in 2013 the first human pluripotent stem cell-based brain organoids, we can start trying at least. So brain organoids, what they are 3D self-assembled structure that faithfully recapitulate some aspect of cellular diversity and tissue architecture of the human brain. The field of stem cell technologies has put a tremendous effort in the development and the optimization of this system. We are nowadays able to derive brain area-specific brain organoids even, and they have been used currently to address a number of questions related to brain-related disorders. Now, regarding outer radial glial cell, we know, thanks to the work of Krixin, what they are supposed to express, how they are supposed to behave, and we know that they are expressing the pan-astroglial marker GFAP, OPEX, and the receptor for LIF. So when we started working with brain organoids, we immediately wondered, okay, are we able to recapitulate an outer subventricular zone, the presence of outer radial glial cell in vitro, using a protocol that we developed based on dual SMAD inhibition that allows us a guide differentiation protocols towards cortical forebrain. Now, what we realized was that it was much more difficult than that. Here you see a zoom in on a ventricular zone enriched by neuronal progenitor cells. As you can appreciate, we are not really able to detect an outer subventricular area outside the intermediate progenitor cells, and those cells are on top of it not GFAP positive. Now, because outer radial glia express the receptor of LIF, and LIF is an activator of the STAT3 signaling pathway, we know that it is extremely required for progenitor cells and stem cell maintenance and expansion. We wondered whether cortical forebrain cells at all would express LIF. And at this point, we went down the rabbit hole because we realized that cortical forebrains don't express LIF. And actually, the lack of outer radial glia was a common feature in many published guided differentiation protocols out there. So then we decided to add exogenous LIF to our culture, and in this context, we were finally able to recapitulate this presence of an outer radial glia, of an outer subventricular zone that was also GFAP positive. Now, single-cell RNA sequencing and comparison between our control organoids and the LIF-treated organoids actually demonstrated that those putative outer radial glial cells are indeed outer radial glia, so they are transcriptionally similar to fetal outer radial glia that has been characterized by Krixin and colleague. And now you might wonder, nice, you are able to derive a primate-enriched progenitor population. What does it mean for the brain microenvironment? Well, by performing velocity pseudo-time analysis, we realized that LIF-treated organoids were showing the emergence of a population, of a neuronal population that was developmentally more mature and not present in control organoids. And this was reflected later on the physiology of the neuronal population and neuronal maturation. So with this, I like to conclude that we are now reaching a moment where we are able to derive a cortical forebrain organoid, derived also by outer glial cell. We have boosted maturation in vitro, and I didn't have time today to show you, but we are also working on approaches to accelerate the differentiation in vitro of astrocytes. We are also able to add human pluripotent stem cell-derived microglia, and also importantly, what I didn't have the time to show you today, the lack of leaf that is not actually produced by cortical organoids and that has to be added exogenously can be compensated by increasing the complexity of the brain microenvironment and by adding, for example, cells of the brain vasculature that are actually producing leaf. And now we are excited to use this model to address some questions on cancer biology, address a question on the microenvironment of brain metastasis, but also of CNS lymphoma. As I mentioned to you, we can recapitulate brain organoids that are even patient-specific by reprogramming fibroblasts or T cells from specific patients. And as a proof of concept, and these are really preliminary data, what we tried to see whether we were able to use those organoids to even model organ tropism in the case of CNS lymphoma. So we used two different lines. One that is an ABC diffuse large B-cell lymphoma of cluster 5, which presented genotype positive for MYD88, CD79B, which is frequently observed in CNS lymphoma, and another cell line that is GCB diffuse large B-cell lymphoma. We added them on the surface of the brain organoids, incubated them for 2 weeks, and checked what would happen to these cells. And we were happy to see that indeed ABC diffuse large B-cell lymphoma of cluster 5 were far more efficient than GCB lymphoma to diffuse and survive in the human brain microenvironment. Now, you might not be completely surprised by this because this is something that you frequently see in the clinic. However, for us, this is exciting because it's giving us a tool to actually address the mechanism that regulate organ tropism and later on cellular adaptation, cellular crosstalk, and niche remodeling. And with this, I'd like to thank all our collaborators, in particular the Studer Lab in New York, that is pushing with us the organoid project that we have right now in revision that I presented to you today. The Bertoni Lab, in particular Luciano Cascione and Afua Mensa, that are on board with us for the CNS project. All our young lab, in particular Antonietta and Raffaele, that are really spearheading the brain organoid projects in the lab, our support and funding agency, and of course all of you for your attention.
Thank you, Arianna, for this beautiful talk. There are questions? Question from the audience? Otherwise, I will ask you a question if I can, right? So if I'm able to. So what about the microenvironment? So you mentioned, so are you able to add like some component of like microglia, whatever, into this complexity of the brain organoid? And whether you have tried to use already this model for drug screening, for instance, and how you are going to screen for that, like so.
Yeah, that's an excellent question. So right now we are definitely able to add microglia. So microglia is coming from a mesodermal progenitor cell, so has to be separately produced, but it can be added to the brain microenvironment, and is currently something that we are doing. What we are planning to do right now is actually a genome-wide CRISPR screen to define how. We are going to do it specifically in the case of melanoma metastatic cells, but it can be done for other brain-related tumor cells, how cancer cells are actually able to remodel the activation of microglia and induce a reactive state that is actually protomeriogenic. So we are very excited to check these. Microglia is known to secrete several cytokines that are actually pro-proliferative. So we are excited to define how the cancer cell is able to instruct the microglia and whether we are able to block that.
Can you use this model for drug screening also? I mean, it's very complex to arrive to a mini brain.
It's very applicable. Yeah, so it's very easy to scale it up. What we're planning to do is to have different approaches. So we are interested not only in how the cancer cell adapt, but really how the niche is remodeled, and we can perform potentially screen for every cellular compartment. So this is definitely work ongoing. Yeah.
Thank you. Well, if there are no further questions, I'm going to the podium, and I thank Arianna to conclude the meeting. So, I mean, if Sylvia allowed me to close the meeting, I think that it's been really a pleasure for me to host you here today. It's been an interesting forum. We have learned how we can use this innovative methodology to study cancer biology and also to deliver novel therapy. And since there is Hans Clever here, I really hope that some of this approach will be used in the near future to speed the process of drug approval, as René did. I mean, in my experience, I'm not young anymore, but it took me 8 years to go from the lab to phase 1 to trial just with a single combination. So it was really painful, and I would like to do this for my next 20 years in a much faster way and modality. So I think that your role will be really instrumental to help us in trying to bring to the clinic innovative combination of drug, like the one that has been presented by René, that is always amazing us with all this amazing idea, a beautiful concept. So by saying that, I thank you for your attention and see you in 2 years, right? So when we'll have the next forum. Thank you very much.